So I'd like to thank the organisers for inviting me to speak at this event. It's a real pleasure to be able to share the work in my lab. So my lab's interested in three sort of fundamental things. The first is to try and develop a really good understanding of how genes are regulated. And then what we try and do is to leverage that to understand how genetic variation leads to disease. And then the final piece is to then try and use that to then go on to develop genome editing strategies to treat patients. And our sort of home turf is the globin loci in erythropoiesis. And this is partly a historical thing. I did my PhD in Doug Higgs's lab. He's worked on alpha globin for the last 30 to 40 years, and it truly is one of the best worked out genes in the genome. And that gives us very solid territory when we develop new technologies to understand what our assays are showing us. But of course, next generation sequencing assays allow us to do genome-wide screens, and we can potentially assay thousands of genes simultaneously now. Single cell technologies allow us to extend out to multiple cell types, and so we're no longer just simply interested in red cells and alpha globin, but we're interested in all the genes in the genome across the entire of haematopoiesis. And then there's a sort of final dimension to this, which is we're really interested in this question about sequence variation. And of course, the large-scale sequencing projects from both Genomics England for rare disease and UK Biobank for common disease allow us to add this additional dimension to try and study what the effects of sequence variation are across all of the different cell types at all of the different genes in the genome. And so one of the key things that we like to think about is how the genome is interpreted by cells. And so 3% of the genome is pretty straightforward, actually. Initially, work done in the 1960s, and you can see here the key work from the lab book of Nirenberg and Mate, literally done with pen and paper, working out that it's a triplet codon code, and it's helped by the fact that it's read by the ribosome in a strict linear order. And then the final thing is that makes this straightforward is there's been very little change in 3.5 billion years of life. So you and I encode our proteins in exactly the same way as archaea, which are some of the oldest organisms on the planet. So what about the remaining 97% of the genome? Well, that's an awful lot more challenging. But it's equally important because over 90% of common disease-associated polymorphisms are found in the non-coding genome. And at the moment, we don't really know what the importance of polymorphisms in the non-coding genome is in rare disease. And the reason this is so challenging is that the non-coding genome is read by transcription factors. And these are proteins which have got very large disordered domains, and so we don't really understand how they work. They bind at sequences called enhancers, which are between 200 and 400 base pairs across, and you get combinations of transcription factors binding at these individual sites. And about 10% of all human genes encode transcription factors, and every cell type at every different stage of development and differentiation is expressing a different repertoire of transcription factors. And effectively what this means is that they're using a different language to interpret their genome. So there's a different level of complexity here. The other thing that makes things challenging is that there's very heavy cell type specific annotation of the genome, both through direct modification of the DNA itself, but also through modification of the histone proteins that the DNA is wrapped around. And then the final challenge is something that we're really interested in my lab, which is that genome structure is a key determinant of gene expression. So this is a super resolution microscopy image here. And you can see down at the bottom left-hand corner, that tiny little red spot is one of the globin genes. And what this means is that the DNA is intricately folded inside the cell, and it means that sequences binding to enhancers can affect gene expression over 1 million base pairs away in the linear sequence, and this adds another level of complexity to it. So in my lab, what we have done is develop assays that allow you to define what's contacting individual genes at extremely high resolution. And so if you go back to the globin loci, these are the two genes over here. Deletion of the genes will lead to thalassemia, but equally we have rare cases where deletion of the enhancers also leads to thalassemia. And this is the assay here that we developed in my lab called microcapture-C, and this precisely identifies these binding sites that are controlling the gene expression. And what's interesting is that you can zoom in on this and then zoom in again, and we can get the resolution right the way down to single base pairs. And that allows us to identify precisely where the transcription factors are binding within these enhancers that are controlling gene expression. And since we published this work, we've gone on to develop maps of whole areas of the genome at single base pair resolution. So this is probably the highest resolution image of any section of DNA inside a cell that's ever been produced to this point. And what that's allowed us to do is to move in to try and model this in real detail. And we've collaborated with Rosanna Colapardo at the Clarendon in Cambridge, who's got the most developed molecular dynamics models for DNA and chromatin. So this model has every single base pair of DNA for 3 kilobases, and it's got every single amino acid in all of the histone proteins. And so there are over 25,000 particles being tracked in this video. And this took us over a week to generate on the supercomputer here in Oxford. And you can see, I don't know if this is projecting that well, but these maps that are generated purely by the biophysics that we programmed into it replicate our maps of high level of resolution chromatin. So how does this allow us to understand how genetic variation leads to disease? Well, the most famous example that we've worked on was in COVID-19. And very early on in the pandemic, there was a region identified on chromosome 3 that was a mystery. And this was really important because it doubled the risk of severe COVID, and it was really common. So about 15% of the population in Europe carry this high-risk variant of the gene, and incredibly common in South Asia. So over 60% of people with South Asian ancestry have the high-risk haplotype. Now, the first thing that we did was try and solve this sort of catch-22 situation that you end up in with these types of genetic variants, where you don't know the cell type and you don't know which specific variant there is in a kind of block of linkage. And for this, we collaborated with Jim Hughes, who'd been developing machine learning models to predict the effects of sequence changes on gene regulation. And he's developed a model that's got 4,000 different cell types in it that allows you to flip in all of the potential variants in the haplotype. And what this showed is that this one individual SNP here is a single base pair change, RS177130554, has an effect, and it has the effect in the lung. We were expecting this to be an immune phenotype. And so the next question was, well, we found the SNP, but which gene is it controlling? And this is the locus. You can see there are 28 SNPs in the haplotype, and there are about 14 genes within 1 million base pairs. And so then we turned to microcapture-C, and we captured from that particular variant, and it identified one gene, and one gene only. And this is a gene called LZTFL1. At that point, it was virtually unstudied. There are about 30 hits on PubMed. And I don't have time to go into it today, but we think that this works through persistent expression of ACE2, which makes pulmonary cells more susceptible to the virus. So what relevance does this have to rare disease? Well, these data are really hot off the press, have only been generated in the last few weeks. But what we've gone on to do is to do microcapture-C experiments. And the way that the assay is set up, we can study hundreds to thousands of genes simultaneously. And we've done this from the 500 or so genes associated with primary immunodeficiencies. And what we're hoping we're going to be able to do is to pinpoint all of the enhancers that might be leading to disease in these patients. And we're really excited to try and integrate these data with Genomics England and other data banks from whole genome sequencing. So finally, I just wanted to talk about developing genome editing strategies to treat patients. And I think Fyodor touched on this yesterday. This is an incredibly exciting time for people who've worked in the globins, because we've now got a CRISPR edit that works for thalassemia and sickle cell disease. It's been approved by regulators, and for thalassemia, it's been approved by NICE in the last month. And it's worth bearing in mind that this has only been developed off the back of 40 years of work to get a deep understanding of how this gene is regulated. And the way that it works is all to do with a protein called BCL11A, which was discovered from a genome-wide association study by Swile while she was working at King's. And this gene, the very tragic thing in these patients is that they all have a normal fetal hemoglobin gene. It's only the adult gene that's generally abnormal in the vast majority of patients, and this is switched off within the first year of life. And what CRISPR does is it targets the expression, and specifically the erythroid-specific expression of BCL11A, turning that gene off and allowing reactivation of fetal hemoglobin. And I'm going to re-show the swimmer plot that Fyodor showed yesterday, this time for thalassemia, just to make the point that it also works just as well for thalassemia as sickle cell disease. These are all the transfusions of the little dots before the treatment, and virtually all of the patients are transfusion independent at the end of the study. And they also have normal levels of hemoglobin, all coming off the fetal gene. But this is the first generation of these products, and actually there are several other strategies that are being pursued, and one of these we have developed ourselves. So there are several different flavors of genome editing, and the one that we really like is base editing. And we got latched onto this very early, so we've watched the original papers come out, and in the early days we were really excited about this technology. And the reason is that if you think about it chemically, what we're able to do is to knock the amine group off a cytosine and convert it to a thymine, and knock the amine group off an adenine and convert it into a guanine. And this is a very chemically simple thing to do. And we started off actually developing an enhancer editing strategy with conventional Cas9, but then we worked out that we could use this to treat a form of thalassemia called hemoglobin E beta thalassemia. So this is the commonest cause of thalassemia worldwide. It's actually quite rare in the UK, but there are about 20,000 births a year. And in parts of the population in Southeast Asia, 60% of the population are carriers because it's highly protective against malaria. And what we found is that we could edit this as a really good target for a base editor, but it would likely send it back to the wild-type sequence some of the time, but it would also send it to a very, very rare variant of hemoglobin that's described in a single family in France. And thankfully, someone published this family, and we know that they have completely normal hemoglobin production. And so what levels of editing can we get with this? Because it's got a canonical PAM sequence, we can get near complete editing efficiencies with this using messenger RNA. So 98% of the edits in some of the best examples are corrected to either wild type or the Orbenus sequence. And when we spotted this edit, we had a pretty good afternoon in the lab actually, because we also spotted that you could use the same trick to fix sickle cell disease. And so for sickle cell disease, you can't send it back to the wild type sequence with a base editor, but what you can do is to convert the A on the non-coding strand to a G, and that changes it to hemoglobin Mercasser. And it's a lovely bit of I love the fact that this was initially published back in 1970 in a minor publication in Biochemica Biophysica Acta, which was never cited. And we found this paper, and it enabled us to realize that this would be safe because this person has a normal phenotype if you have hemoglobin G Mercasser. And what's wonderful is we licensed this technology to Beam Therapeutics. They have then gone to the trouble of actually evolving a new editor to specifically make this edit with high efficiency. And so this is potentially what is possible in this space. Just briefly to touch on off-target effects, the point I just want to make with this graph is that the vast majority of off-target effects that you can predict, both in silico and in vitro, lie in the non-coding genome. And one of the things we've been interested in doing is repurposing the machine learning that we've been using to try and identify the effects of sequence variants to look to see what the effects of off-target effects might be. And then just finally to kind of head into the jammy dodger country. So genome editing for other indications. So one of the things is that when we're editing for sickle cell disease, what we're actually editing are the long-term haemopoietic stem cells. And of course, when you do that, you edit all of the other cell types that come out of haemopoietic stem cells. So that's the entire immune system and platelets. And of course, as people have mentioned multiple times in this conference, we can then simply use the entire process that's been developed actually over decades for bone marrow transplantation, but combined with editing, and simply change the guide RNA of the editor. So we've been very interested in systematically identifying editing strategies for rare disease, and here we're thinking about what's required for the patient, and that will be dependent on cell type, editing efficiencies, and so this is a clinical piece, and then what's possible. And we've looked at this for all of the genes associated with immunology and haematology phenotypes in Genomics England, and we've mined about 60,000 variants out of ClinVar. And we think over half of those are potentially fixable today with an editing strategy. Of course, this will fall if you actually go in vivo and check that these edits work and then look for off-target effects. But this is a potentially really important space, and the editing technology is only going to improve over the next few years. And that just leaves me to thank all of the people in my lab who've worked incredibly hard on this these projects and all of my collaborators.